Every needle that's ever gone into your arm was checked first. Checked by an animal with bright blue blood, older than trees. A gallon of it sells for $60,000. So every year we catch 500,000 of them, drain a third of it, and put them back. In 1956, a researcher named Fred Bang lifted a dead horseshoe crab out of a tank at Woods Hole, Massachusetts. Its blood had turned to jelly. Every drop of it solid. So what turns an animal's whole blood supply into a lump? Bang was a pathobiologist at Johns Hopkins who spent his summers at the Marine Biological Laboratory. His wife Betsy was a scientific illustrator, trained to notice the things other people walk past. He'd been injecting sea creatures with bacteria to see what their immune systems would do about it. Most of them did very little. The horseshoe crab did something nobody had a name for. Start with the color, because that's the part people get wrong. Our blood runs on iron. Hemoglobin grabs oxygen, the iron oxidizes, and you get the color of rust. The horseshoe crab runs on copper instead. The molecule is called hemocyanin, and the moment it meets air, it turns a pale, cloudy blue, less like blood than like milk stirred into ink. And it carries exactly one kind of cell. One. No white cells, no antibodies, no immune memory, none of the machinery you'd expect. The cell is called an amebocyte, and under a microscope, it looks like somebody packed it with sand. Those grains are parcels of a protein called coagulogen. Bang kept pulling the thread. Only one family of bacteria set the reaction off. The gram-negative bacteria, the group behind pneumonia and meningitis. Then he heat-killed the bacteria and injected them dead. The blood clotted anyway. That result is the whole story in one line. The crab wasn't reacting to something alive. It was reacting to a poison those bacteria carry in their outer wall called endotoxin. And endotoxin keeps working long after the bacterium that made it is gone. So the clotting wasn't a wound closing. It was a quarantine. An animal with no immune system worth the name had exactly one move available to it. Find the invader, wrap it in gel, stop it traveling. And it could find that invader in amounts so small nothing in a hospital lab could match it. Bang wrote it up in 1956. Then, for reasons nobody has ever fully explained, he put it aside. Why would anyone pay a fortune for a fluid that comes out of a beach animal? Because of what it replaced. Before this, if you made an injectable drug, you tested it on rabbits. Not one rabbit, colonies of them. Thousands of animals kept in temperature-controlled rooms for that single purpose. You injected your product into the rabbit, put a thermometer in it, and waited to see whether it ran a fever. It worked, roughly. It was also slow and expensive, and a rabbit is a living animal rather than a measuring instrument. Two of them given the same dose don't always agree. In 1963, a colleague at Johns Hopkins told Bang he was never going to crack this on his own. That was Dr. C. Lockard Conley, who ran the hospital's hematology division, and his advice was simple. Get a blood specialist. He recommended a research fellow from his own lab called Jack Levin. Levin later described it as two professors at the same school talking to each other. That conversation is the reason any of this exists. Levin's contribution was the mechanical part, and it mattered enormously. He worked out that the whole reaction lived in the amebocytes, which meant you didn't need a crab at all. You needed the cells, wash them, break them open, throw away the wreckage, and keep the liquid that pours out. They called it Limulus amebocyte lysate, LAL. Levin published the description in 1964, 8 years after Bang first saw the jelly. Here's what it does, and the elegance of it is that a technician can read the answer across a bench. You put a drop of your product into a tube of the stuff and hold it at body temperature. Then you pick the tube up and turn it upside down. If a solid gel has formed, it stays put, and that batch is contaminated. If it runs down the glass, you are clean. That's the test. Invert the tube and look. The sensitivity is where it stops being a laboratory curiosity. LAL registers endotoxin at parts per trillion. a sugar cube dissolved in a reservoir. And it takes about an hour against a rabbit colony you had to feed, house, and watch for days to get a fuzzier answer. The United States Food and Drug Administration licensed it in the 1970s. Within a few years, the rabbit rooms were closing. So how does a fluid get to $60,000 a gallon? The number comes from a widely reported price of about $15,000 a quart. The companies do not confirm their pricing, so treat it as the industry's reported ballpark rather than a receipt. Even so, a gallon of it costs roughly what a new car costs. What you are buying is the last checkpoint before something enters a human body. Think about what that covers. Every vaccine, every bag of IV saline, insulin, chemotherapy, dialysis fluid, anesthetic. Then everything that gets implanted and stays. Pacemakers, stents, artificial hips, replacement knees, surgical mesh. All of it cleared with this, or with something built from it. And the reason ordinary sterilization is not enough is the thing Bang stumbled onto with his dead bacteria. An autoclave kills bacteria. It does not destroy endotoxin. You can run a batch of drug through a sterilizing cycle, kill every living organism in it, and still be left with a solution full of molecular debris that will make somebody very sick. Endotoxin in the bloodstream gets read by your immune system as an emergency. Fever first, then blood pressure crashing as the vessels dilate. Then, in a serious exposure, organs failing. It is a central mechanism in septic shock, and the molecule does not have to be attached to anything living to do it. Nobody publishes a clean global figure for how often this test runs. The estimate that circulates is around 70 million endotoxin tests a year, which you should take as an order of magnitude rather than a count. Even at that rough scale, it means the reaction is happening somewhere in the world about twice a second, permanently. Which is why, when the COVID vaccines were being built at speed in 2020, this test sat right in the middle of the process. The body that sets U.S. drug standards put out guidance for vaccine developers that year because nothing was going into anybody's arm without passing this checkpoint first. The market for the reagent itself is small. Figures around $50 million a year get quoted. A rounding error guarding a multi-trillion-dollar industry. And the price has almost nothing to do with how hard it is to make. It is expensive because of where it comes from. There is no factory. There is a boat, a beach, and an animal somebody has to go and physically pick up. So, what is this thing? Not a crab. That's not a technicality, it's a different branch of the tree. Crabs and lobsters are crustaceans. The horseshoe crab is a chelicerate, which puts it in with spiders, scorpions, ticks, and mites. It has no antennae. The first pair of appendages by its mouth are chelicerae, the same structures a spider uses. It's closer to the thing in the corner of your garage than to anything you'd order in a restaurant. The spiked tail isn't a weapon either. There's no sting in it. It's a lever. The animal uses it to flip itself back over when a wave dumps it upside down on the sand, which is otherwise a death sentence. It has 10 eyes, 2 big compound ones on the sides of the shell and the rest scattered over the body, including a pair on the tail. They're so large and so easy to get an electrode onto that a researcher called Keffer Hartline spent most of a career on them. What he worked out was how any eye sharpens an edge before the signal ever reaches the brain. He took the 1967 Nobel Prize for it. A lot of what we know about how vision works came out of this animal. Its fossil relatives go back to the Ordovician, roughly 445 million years, older than trees. It predates Pangea, which means it watched the supercontinent come together and then break apart again. There's a fossil bed at Luoping in Yunnan province in China that shows how little has changed. The horseshoe crabs preserved there are about 244 million years old, and the detail is fine enough to make out book gills, walking legs, muscle, individual bristles. Put one next to a living animal off a New Jersey beach and you'd struggle to tell them apart. It was already here for the first of the 5 great mass extinctions. It came through the one at the end of the Permian, which wiped out something north of 90% of marine species. It came through the asteroid. 4 species are left. One, Limulus polyphemus, runs the Atlantic and Gulf coast from Maine down to the Yucatan. The other three are Asian, and they're in worse shape. The tri-spine horseshoe crab ranges from southern China up to western Japan. Japan gave it legal protection back in 1928. Taiwan's coastal populations used to be abundant and are now barely seen inshore at all. IUCN moved the species to endangered in 2019, and once a year it does the thing that made it famous long before anyone drew its blood. In spring, on the high tides around the full and new moons, they come out of the water in numbers. The females haul up the sand and dig. The males clamp on and follow. Each female buries clusters of thousands of eggs and goes back to the sea. It survived everything the planet threw at it for 445 million years. Then, a researcher in Massachusetts found out what was inside it. So what happens to a crab that gets collected? There are a handful of licensed facilities on the U.S. Atlantic coast. Charles River Laboratories has run one in Charleston, South Carolina for about 30 years. Lanza operates a site in Walkersville, Maryland. Associates of Cape Cod is the third big name. The animals come in by truck, trawled, or hand-collected off the beaches. They're washed down to clear the sand and shell debris. Anything with visible damage gets pulled out. The rest are bent backwards over a rack in rows, the joint between the two halves of the shell opening as they flex. A technician puts a large gauge needle through the membrane at that hinge, straight into the heart. The blue drains out into a bottle underneath. On the rack, a crab looks like it's been folded in half and left there. They take up to 30% of the animal's blood volume. Hold on to that number. Then, within 72 hours, they're trucked back and returned to the water, usually near where they were picked up. That window is a regulatory requirement rather than a courtesy. Leave them out of the water longer and the losses climb. Blood volume recovers in about a week. The cell count is the slow part. The amebocytes, which are the only thing anybody wanted, take months to rebuild, and a crab short on amebocytes is a crab with a weakened immune response in an ocean that's essentially a bacterial broth. They can legally be caught and bled more than once in a season. The scale is bigger than most people assume. A U.S. Geological Survey paper covering 2013 to 2017 puts it at 525,000 crabs a year captured and bled. The Atlantic States Marine Fisheries Commission, which manages the fishery, ran an average of about 417,700 a year across a longer window going back to 2004. So, catch it, drain a third of it, drive it back, drop it in the surf. For years, the industry's position on what that does has been consistent. Under 3% of them die. That number is the most fought-over figure in this whole story. So how many actually die? Depends entirely who's counting. The biomedical companies have historically said 3% or lower. The Atlantic States Marine Fisheries Commission doesn't accept that. For management purposes, it applies 15%, with a plausible range running anywhere from 4 to 30. Independent studies have landed in the upper part of that range. Run the commission's number and you get roughly 61,500 crabs a year de-wying from the biomedical process along the Atlantic coast. The U.S. Geological Survey figure for more recent years is about 78,750. A filled football stadium every year from bleeding alone. Then in 2020, a team led by David Smith at the U.S. Geological Survey did something nobody had managed before. Instead of running crabs through a lab and watching them for a few days, they went to the U.S. Fish and Wildlife Service tagging database. Years of individually tagged animals, released and later recaptured. They compared bled crabs against unbled crabs tagged in the same years, in the same places. If bleeding is quietly killing 15 to 30% of them, the tag should show it. Bled animals should come back at lower rates, year after year, fewer and fewer. They didn't. Survival wasn't lower for bled crabs. In several comparisons, the bled ones came out slightly ahead. The authors are careful about what that means, and it's worth being just as careful. Biomedical facilities cull the injured ones before bleeding and prefer healthy adults, so the bled group may simply have been a fitter set of animals to begin with. That's their caveat, not a critic's. But the same analysis turned up something else, and this is the part that doesn't get repeated enough. The recapture probability dropped after bleeding. The tagged animals were seen again less often, and it showed up more strongly in the males. Recapture happens on the spawning beaches. If a crab stops being recaptured, the most likely reading is that it stopped turning up to spawn. So the needle may not be killing them the way everyone assumed. It may be doing something quieter. Take a third of an animal's blood and it keeps living. It just stops going to the beach, which matters enormously because of what's waiting on that beach. So, who else needs a horseshoe crab's eggs? Delaware Bay, mid-May. The crabs come ashore on the high tides and bury their eggs a few inches down. Then the next wave of crabs digs up the last wave's nests and storms turn the sand over. And what you end up with is a layer of loose eggs sitting on the surface of the beach. Soft, green-gray, no shell to crack and nothing to chase. Almost pure fat. Larry Niles has been standing on those beaches for more than 25 years. He used to run New Jersey's Endangered Species program and works independently now. And for most of that time, his job in May has been to trap, weigh, band, and count the birds that show up for those eggs. The one that matters most is the red knot. The Rufa subspecies specifically, a shorebird about the size of a robin, rust-colored across the chest in breeding plumage. It winters at the bottom of South America, in Tierra del Fuego, and breeds in the Canadian Arctic. The trip runs to something like 9,000 miles each way, one of the longest migrations of any bird alive. In a good year, 80 to 90% of the entire Rufa population is standing on Delaware Bay inside the same fortnight. They arrive nearly empty. They've got about 2 weeks to roughly double their body weight before the last leg north, and Niles' team has a number for it. Around 180 grams by the last week of May. Hit it and you can breed. Miss it and you can't. One bird made all of this legible to people who'd never heard of any of it. He was caught and banded in 1995 and carried an orange flag reading B95. Most red knots live 5 or 6 years. He kept coming back. Over the years, biologists worked out that the distance he'd flown in his lifetime was further than the distance to the moon, and they started calling him Moonbird. He was still being sighted into his 20s. There's a statue of him at the DuPont Nature Center in Delaware. He was an outlier. What happened to the rest of them isn't. In 1986, the count on Delaware Bay was around 93,000 red knots. Through the 90s, it held above 80,000. By the turn of the century, it was down near 30,000. One recent count came in at 22,000. In 2021, a survey found 6,800. Egg densities on the New Jersey spawning beaches have dropped more than 90% since the 1980s. And in 2020, the bay showed exactly how thin the margin is. The water stayed cool through the spring and the spawn ran late. Storms churned the beaches. The knots arrived on schedule because they leave South America on a calendar written into them, and they landed on sand with almost nothing in it. Here's the cruel part. Almost none of them die on Delaware Bay. They leave underweight, and they die 3,000 miles later over open ocean or out on the tundra, somewhere with nobody counting. So if it isn't the needle, what is it? Bait. Horseshoe crabs get chopped up to catch two things: American eel and whelk, which you will see sold as conch. A crab in a pot brings in animals worth more per pound than the crab is. That is the whole economics of it. Look at what happened in the 90s. In 1992, the Delaware Bay harvest was under 100,000 crabs a year. By 1997, it was over 2.5 million. 5 years, and the take went up more than 20-fold, and the egg layer on those beaches collapsed behind it. Put the numbers side by side, and the proportions are obvious. The USGS puts biomedical mortality at under 13% of the up to 1 million crabs landed coastwide each year. The rest is bait. The industry paying $60,000 a gallon is the smaller pressure on this animal by a wide margin. Niles and others spent years pushing on it, and New Jersey banned the harvest outright in 2008. In 2015, the federal government listed the Rufa red knot as threatened. The management that came out of all this is genuinely unusual. The Atlantic States Marine Fisheries Commission, the U.S. Fish and Wildlife Service, and the USGS built something called the Adaptive Resource Management Framework, and it does what fisheries rules almost never do. It sets how many crabs can be taken partly according to how the birds are doing. 2 species, 1 quota. There is a detail in there people find hard to place. The biomedical companies have helped pay for the trawl survey that framework runs on. Charles River, Lanza, and Associates of Cape Cod together pledged over $100,000 toward it in a single year. The industry taking the crabs is helping fund the count that constrains it. You can read that cynically or generously, and both readings hold up. There is also a real disagreement here, and it belongs on the table. Delaware's environmental agency has publicly rejected the idea that crab eggs are the limiting factor for the red knot. Their position is that egg abundance in the bay is sufficient and that the birds are being hit by climate problems up on the Arctic breeding grounds instead. The timing of snowmelt, the insects that come with it. Niles' data says the birds are leaving the bay underweight. Both things can be partly true, and the argument is not settled. What is not in dispute is which pressure has an exit. Nobody has invented a replacement for an eel pot. The pharmaceutical industry is a different case. It has had an alternative sitting on the shelf for a very long time. So there's a synthetic version of this. Why is anyone still bleeding crabs? The science was finished in 1997 at the National University of Singapore by Jeek Ling Ding and Bo Ho. Their insight was that the crab's clotting cascade is a chain, and only the first link does the detecting. That first protein is factor C. It's the sensor. Everything downstream of it is amplification, machinery for turning a signal into a gel you can see. So you don't need the cascade, you need the sensor. Ding and Ho cloned factor C and produced it in cell culture. Recombinant factor C. No crab anywhere in the process. It came with advantages beyond the obvious one. Animal-derived reagents vary between batches because animals vary. A recombinant protein doesn't, and your supply stops depending on how many crabs washed up on a beach in South Carolina that season. Eli Lilly moved to it in 2016 and has used it since. Then it sat. In 2020, the U.S. Pharmacopeia put out a notice reaffirming its commitment to bringing the synthetic into official standards. On the 26th of July 2024, its Microbiology Expert Committee finally voted to adopt Chapter 86, which sets a formal standard for endotoxin testing with non-animal reagents. The text published that November. It came into effect in May 2025, 1997 to 2025. 28 years between the thing existing and the thing being recognized, which is the same shape as the start of this story, scaled up. Bang saw the reaction in 1956, and it sat in a drawer for 7 years until somebody put the right 2 people in a room. The replacement for that discovery was cloned in 1997 and sat for nearly 3 decades. Discovery moves fast. Adoption doesn't. More than 50 conservation and health organizations pushed for the change through the Horseshoe Crab Recovery Coalition, with backing in Congress from Representative Frank Pallone and Senator Cory Booker, both of New Jersey, the state that shut its own harvest down. One honest caveat, and it's the reason nothing changed overnight. Chapter 86 permits recombinant reagents. It doesn't require them. Every manufacturer still has to validate the switch for each of its own products and prove to regulators that the new method reads its particular drug correctly. That costs money and time, and it carries the risk that something already on the market fails its own retest. For a company whose existing method is approved and working and unchallenged, the incentive to volunteer for that is close to zero. So crabs are still being bled this year. The alternative works. It was never a question of whether it works. So once you know blood doesn't have to be red, is the horseshoe crab actually unusual? Not remotely. Copper-based blue blood is all over the ocean. Octopus have it. So do squid, cuttlefish, lobsters, and a long list of snails. Spiders and scorpions run on it too, which stops being surprising the second you remember what family the horseshoe crab belongs to. And down there, it isn't a quirk, it's the better engineering. Hemocyanin outperforms hemoglobin in cold water with low oxygen, which describes most of the deep ocean. In that world, blue is the correct answer and red is the odd one out, which makes our version of the phrase almost perfectly wrong. We call aristocrats blue bloods from the Spanish sangre azul, an old idea about noble families being finer, their veins showing through pale, untanned skin. Real blue blood was never in a palace. It was in the tide pools, in the animals nobody wanted to touch. Then it gets stranger. In the forests of New Guinea, there are lizards whose blood is lime green. The genus is Prasinohema, and the green isn't confined to the blood. Their bones are green. Their muscle is green. Their tongue and the lining of their mouth are green. The cause is a pigment called biliverdin, which your own body makes when it breaks down old red blood cells and then works hard to get rid of, because in quantity it's toxic. It's the green you see around the edge of a healing bruise. These lizards are carrying it at concentrations 40 times the level that would kill a person, and they're completely fine. Christopher Austin at Louisiana State University has spent decades on them. His team found the trait evolved at least 4 separate times in different lineages, which means something about it is worth having. The leading guess, from the parasitologist Susan Perkins, is defense. A related pigment is toxic to malaria parasites, and these lizards get malaria too. Nobody has proved it. The green lizard is still, at bottom, unexplained. And then there's the animal that gave up on the whole idea. In 1928, a Norwegian biologist called Ditlev Rustad hauled a fish out of the water off Bouvet Island in the Antarctic. Scaleless, ghost white, half see-through, he cut it open and there was no red anywhere. The gills were pale as yogurt. The blood ran clear, like water. He had found the ice fish. 16 species in the family Channicthidae, and they're the only vertebrates on this planet with no hemoglobin at all. Somewhere around 8.5 million years ago, the gene broke. And in water that cold and that rich in dissolved oxygen, it didn't matter. They absorb oxygen straight into their plasma. To make up for it, they run oversized hearts, more blood volume than a normal fish, and antifreeze proteins that latch onto ice crystals and stop them growing. A backboned animal, alive and hunting, with clear blood. So line them all up by price. Human blood is worth roughly $1,500 a gallon. The horseshoe crab's runs about $60,000. King cobra venom is quoted near $153,000. And the most expensive liquid on Earth is the venom of the deathstalker scorpion, somewhere in the region of $39 million a gallon, a figure that only really exists on paper, because you'd have to milk one scorpion about 2.6 million times to fill a jug with it. The horseshoe crab doesn't top that list. It's the only thing on it that has protected everyone. So go back to the tank in Massachusetts, and the dead crab, and the blood that had set like jelly. What Bang was holding was 445 million years of solution. No antibodies, no immune memory, one cell type and one move. Find the invader, wrap it where it stands, don't let it travel. It was enough to carry an animal through 5 mass extinctions, an asteroid, and the assembly and destruction of a supercontinent. We took that move and pointed it at ourselves. Every needle that's ever gone into your arm passed a checkpoint built out of it. And the animal is going the way it does everything else. Quietly. It doesn't wash up in piles. There's no carcass on a beach for anyone to film. It just stops arriving. Fewer on the sand each May, fewer eggs in it, and a bird from Tierra del Fuego landing on an empty beach with 3,000 miles left to fly. Larry Niles has been asked more than once whether he saved the red knot. He won't say yes. What he says is that he slowed the fall. Every May, he's back on the same beaches with a clipboard, waiting to see how many come back. If you want to know what else is quietly running out while nobody films it, there's a whole shelf of these. Start with the next one.